Everyone, today's topic is Harshey cheese, Harshey and cheese, Harshey and cheese experiment, transduction experiment, transduction experiment. To prove DNA is genetic material. In our previous lectures, we discussed two experiments. Number one experiment was Griffith's transformation experiment, and every McLeod and McCarthy experiment, which gave a clue that DNA is the genetic material. But the best proof regarding this long pending problem, that which is the genetic material in living organisms, whether it is proteins, whether it is RNA or DNA, this was once and for all settled by the best experiment conducted by Harshey and Chase in 1952. This experiment is also called as Harshey and Chase transduction experiment or transduction principle. Let's now first discuss what is transduction. Students, in bacteria, true sexual reproduction is not present. Okay? What does that mean? That means that bacteria don't possess sex organs. Bacteria don't produce male gametes, female gametes. There is no fertilization, there is no formation of zygote whatsoever. But in bacteria, a part of DNA is transferred to another bacteria. A part of DNA is transferred from donor bacterial cell to the recipient bacterial cell, right? And we say that this genetic exchange or this genetic recombination is a type of sexual reproduction which is shown by bacteria, though a true sexual reproduction, which you might have learned in angiosperms, flowering plants, I mean to say, other living organisms, where there is formation of sex organs, where there is fusion, formation of gametes, formation of zygote. Unlike those, living organisms, bacteria don't show true sexual reproduction. They show a sort of genetic recombination, gene recombination. What does that mean? That means a fragment of DNA is passed from donor bacterial cell to the recipient bacterial cell. And this genetic recombination in bacteria takes place by three methods. Number one method is transformation. Which was experimentally proved by Griffith in his experiment in 1928. And now, another method, that is transduction. This transduction method of genetic recombination in bacteria was proved by Harshey and Chase Alfred Harshey and Maratha Chase in 1952. What is this transduction actually? Let me define this transduction. It's a special type of genetic recombination in which fragments of DNA are transferred from one bacterial cell, that is donor bacterial cell, one bacterial cell, which is regarded as considered as donor bacterial cell, to the another bacterial cell, which is considered as recipient cell, by the agency of bacteriophages. This is transduction. I will repeat it again. Transduction is a special type of genetic recombination in which fragments of DNA are transferred from one bacterial cell, that is donor bacterial cell, that is regarded as donor bacterial cell, to the another bacterial cell, which is considered as recipient bacterial cell, by bacteriophages or phages, whatever pronunciation you like. This is transduction. Let us see what are these bacteriophages. You know, we are discussing Harshey and Chase experiment, which is also called as Harshey and Chase transduction experiment, to prove that DNA is the genetic material. So, before going into the details of Harshey and Chase experiment, we are just trying to learn what does transduction actually mean. I told you that transduction is a special type of genetic recombination in which fragments of DNA are transferred from one bacterial cell, which is also called as donor cell, to other bacterial cell, which is also called as recipient cell, by bacteriophages. It's the point which I want to tell you. What are bacteriophages? You know, viruses are specific in their action. Those viruses which infect the plants can't infect the animals, and those viruses which infect animals can't, infect plants. And viruses are the only, microorganisms, I would say, which have the ability to attack and infect bacteria also. And I may tell you, those viruses which attack and infect and kill bacteria are called as bacteriophages. Okay? I hope that you might have got it. Those viruses which attack, infect and kill bacteria, those are known as bacteriophages, or simply phages or phages. These bacteriophages are of different types. There are many types. Okay? One common type which have been extensively studied, those are known as T phages. What are they known as? T phages. T phages are special type of bacteriophages. One point regarding T phases? T phases are most extensively and commonly studied bacteriophages. Bacteriophages. There are many types of T phages, and these T phages occur in a T series, such as T1 bacteriophage, T2 bacteriophage, T3 bacteriophage, T4 bacteriophage, T5 bacteriophage, T6 bacteriophage, T7 bacteriophage. We say that these are T series of bacteriophages. Here you must remember that T here stands for type. Okay? Is that clear to you? I am telling you that bacteriophages are of many types. Most common and extensively studied bacteriophages are the T phages. Okay? These T phages occur in a T series of phages, such as T1, T2, T3, T4, 5, 6, and 7. Here T stands for type. Okay? Now, all these bacteriophages, these T7, from T1 to T7, these have been divided into two groups. Okay? One, T-even bacteriophages, T-even bacteriophages, and T-odd bacteriophages, T-odd bacteriophages. So, I am telling you that all the bacteriophages, the series of T series bacteriophages, they have been divided into two types, T-even bacteriophages and T-odd bacteriophages. Okay? Let me tell you T-even bacteriophages. T2, okay, T4 and T6. These are placed into the group of T-even bacteriophages. T-even bacteriophages. These bacteriophages are again most commonly and extensively used bacteriophages. Okay? These phages have a simple structure. They have a head and a contractile tail. Their structure we will discuss in detail after some time. Okay? But I am telling you that these have a simple structure. Almost all the viruses have a simple structure. They have just head and a tail. Okay? But this tail is contractile tail. Contractile, which can contract because it is made up of contractile sheet of proteins. Okay? And these T-even bacteriophages, okay, they have contractile tail and they have a unique base, that is 5-hydroxy methyl cytosine. These are characteristics of, okay, T-even bacteriophages. T2, T4 and T6 are T-even bacteriophages out of T1 to T7 bacteriophages. Okay? These bacteriophages have a simple structure. They have head and tail. This tail is contractile because it is made up of contractile sheath of proteins, those proteins which are flexible, which can contract. Okay? And these, bacteriophages, T-even bacteriophages, these have a unique base, 5-hydroxymethyl cytosine. This is T-even bacteriophage. Now come to the T-odd bacteriophages. T-odd. T1, okay, T3, okay, T7. Bacteriophages are considered as, T-odd bacteriophages. Okay? What about T5? I will tell you after some time. T1, T3 and T7. These are considered as T-odd bacteriophages. And another class is T5 bacteriophages. In fact, I may tell you that these bacteriophages are classified into three groups. One is T-even bacteriophages, which includes T2, T4, T6. Other is T-odd bacteriophages, okay, which includes T1, T3 and T7. And T5 forms a separate group of bacteriophages. Okay. Now, I was discussing T odd bacteriophages. These are T1, T3 and T7 bacteriophages. They are, genetically and serologically different from T-even bacteriophages. They do have structure like T-even bacteriophages, which is that they have a head and tail. But only difference is that their tail is non-contractile. 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 While the tail of T1 bacteriophages is contractile, their tail is non-contractile. Whose tail is non-contractile? T odd bacteriophage's tail is non-contractile. I am telling you that though the structure, the appearance, the shape of both T1 bacteriophage and T odd bacteriophage is similar. They look like a tadpole. They look like a spermatozoid. They have a head and tail. But the tail of T odd bacteriophage is non-contractile, while the tail of T-even bacteriophage is contractile. What does that mean? I told you that their tail, T-even bacteriophages, have a tail which is made up of contractile proteins, which consists of contractile proteins, whereas the protein is present in the tail of T-odd bacteriophages are not contractile proteins. Okay. And one more difference between T-even bacteriophages and T-odd bacteriophages is that T-odd bacteriophages have a normal cytosine base. Their base is not 5-hydroxymethyl cytosine. It is just a normal cytosine. Okay? Now, what about this T5 bacteriophage? T5 bacteriophages form third group of bacteriophages only because of one difference with T-odd bacteriophages. They have similarities. T-odd bacteriophages do have cytosine base, and T5 bacteriophages also have cytosine base. But only difference is that the T-odd bacteriophages have short non-contractile tail, while as the T5 bacteriophages have long non-contractile tail. Okay, this is their classification. Now come to the out of these all bacteriophages, T2 bacteriophage. Okay, T2 bacteriophage is most common bacteriophage. Let us discuss the structure of this T2 bacteriophage. Okay, T2 bacteriophage. Structure of T2 bacteriophage. T2 bacteriophage. T2 bacteriophage. T2 bacteriophage. Okay, number 1 point is that T2 bacteriophage belongs to T-even group of bacteriophages. T-even group of bacteriophages. Okay, this is 1 point. T2 bacteriophages have a tadpole or spermatozoid. Okay, like appearance. This T2 bacteriophage has a tadpole or spermatozoid-like appearance. It has 2 parts. It has 2 parts. Number 1, head. Number 2, tail. Head is polyhedral. It is polygonal. Okay? It has a protein coat. This is protein coat, which is known as capsid. inside the protein coat or capsid, there is a nucleic acid core. I mean, there is a central part which consists of nucleic acid DNA. But what DNA? You might be knowing that viruses possess nucleic acids. Either they possess DNA or they possess RNA. But it is never seen that a virus may possess both DNA or RNA. Either DNA or RNA. These bacteriophages are those viruses where the nucleic acid or the genetic material is the DNA. This DNA is double-stranded. Okay? Hence, T2 bacteriophage, which has two parts, head and tail, out of which head we are discussing. I'm telling you that this head has a polyhedral shape, which has many sides, which has many faces. I mean to say, this head has a protein coat. This head has a protein covering, which is also known as capsid. Okay? It's made up of proteins. Inside the protein coat, there is a central part. There is a core, which consists of nucleic acids. Which nucleic acid? This is DNA. This is DNA. But you must here remember that in DNA, I am telling you, phosphorus is present, but sulfur is absent. While as in proteins of protein coat, sulfur is present, but phosphorus is absent. Sulfur is present, phosphorus is absent. Where? In the proteins of capsid. While as in the DNA of nucleic acid core, okay, phosphorus is present, while as sulfur is absent. While as sulfur is absent. This is tail. Now, head. This head is connected to a cylinder tail. This head is connected like this, I am telling you. This head is connected to a cylinder, very thin tail. This tail is hollow. This is hollow. It has a sheath of proteins. It is covered by a sheath made up of proteins. Okay? At the end of the tail, there is a structure that is known as end plate. That is known as end plate. End plate has two types of structures attached to it. On its upper side, it has 6. How many? 6. I am drawing just 1 or 2 here. These are actually 6. 6 tail fibers. And on its underside, like this, it has again 6. I am drawing here 1 or 2. It has again 6 tail pins. These are tail fibers. Okay? These are tail pins. Only difference between tail fibers and tail pins is that tail fibers are attached on the upper side of, this end plate, basal plate or end plate, which is the end of the, hollow tail. While as tail pins are attached on the lower side or underside of this end plate. What is the function of these tail pins and tail fibers? When same T2 phage attacks a bacterium, host, okay? It gets attached to the bacterial cell surface, cell wall, by these tail pins and tail fibers. Okay? One more function they also have. These tail fibers and tail pins possess enzymes such as lysozyme, which help us in breaking down the cell wall so that the viral DNA can enter into the bacterial cell. Okay? This is the structure of, bacteriophage T2. Very quickly, I will repeat the structure of bacteriophage T2. It has two parts, head and tail. Okay, head is polygonal shape. It has two structures. It has a central part that is known as nucleic acid core consisting of DNA. Okay, and the central part, which is known as nucleic acid core, which consists of DNA, it is covered by a covering coat known as protein coat, which is made up of proteins. Okay, then this head is connected to a cylinder tail. Okay, this tail is hollow. It is covered by a spiral, sheet of proteins. Okay, at the end of the tail, there is a structure known as end plate or basal plate. This basal plate has two types of structures attached to it. On its upper side, it has long structures, those are known as tail fibers. Whereas on this lower side or underside, I will say, it has tail pins, structures like tail pins. One thing is common between tail fibers and tail pins, that both are 6 in number. But regarding their length, if I will tell you, tail fibers are longer than tail pins. What is their function? During the attack or during the infection, when these T2 phages infect a bacterial cell, these phages get attached to the surface of bacterial cell, to the cell wall of bacterial cell by these tail pins and tail fibers. And one more function of these structures, tail pins and tail fibers, is that they secrete enzymes. One of them is lysozyme. It is because of this lysozyme that they can, in fact, the bacteriophage can break down the bacterial cell. Okay, one more thing which I will tell you. In the middle of, end plate, there is a perforation, there is a pore through which viral DNA can pass on into the bacterial cell. This is the structure of bacteriophage. Okay. Now, come to the, actual experiment, which is our topic today. Before going to the actual experiment, it was necessary to know about bacteriophages, which are very crucial in this experiment. Okay? Harshey and Chase conducted their experiment. That experiment is known as transduction experiment or transduction principle. What they used? They used two types of living organisms in their experiment. One was bacteriophages. What the bacteriophages are? They were to you. I hope that you might be now aware about ABC of these bacteriophages. And for that matter, A to Z about these bacteriophages. Okay? Now, next one is E. coli, bacteria. These were two living organisms which Harshey and Chase used in their experiment. What they used? They used bacteriophages and bacterium E. coli. This E. coli is a very common bacteria. It usually occurs in the intestines of human beings. It performs many important functions there. You might be knowing. Okay? So, because it occurs in colon, this is known as bacillus colon. Colon bacillus. This is known as colon bacillus, I may say. And those bacteriophages which occur in intestines, those are known as coliphage phages. Okay? Now, Harshey and Chase used in their experiment viruses. Which type of bacteria? Viruses? Bacteriophages. Which type of bacteriophages? T2 bacteriophages. Okay? And they also used bacterium. Which type of bacterium? E. coli. Okay? Where this E. coli usually occurs? It occurs in human intestines. Where there? In colon. Okay? Hence, known as colon bacillus. And many bacteriophages which occur in, intestines, those are known as coliphages. Okay, these were two living organisms. Now let us discuss how they performed experiments with T2 phage and E. coli. Okay, let us discuss T2 phage. Now it's now clear to you that two organisms used by Harshey and Chase were T2 phage, okay, and E. coli. They performed experiments. They performed two separate experiments. Okay. Suppose this is now one experiment number 1, okay, and this is experiment number 2. In first experiment, they just, tried to multiply the bacteriophages, T2 bacteriophages. We can't culture viruses without a living host. They prepared, in first experiment, they prepared a culture medium. Suppose this is culture medium. In this culture medium, in addition to other nutrients, in addition to other materials necessary for growth of, bacteriophages, T2 bacteriophage, they placed E. coli into it. Okay? Experiment number 1, culture medium. Besides possessing necessary culture nutrients, culture medium, ingredients, E. coli was also placed in this culture medium. Because viruses, T2 phages, would not, multiply without a living host. Here E. coli was used as a living host. Okay? But one important thing, what they did, Harshey and Chase, they used it here in this, medium, first medium, culture medium, radioactive sulfur. Radioactive sulfur. Radioactive sulfur. What does that mean? That means they used radioactive isotope of sulfur. Which one is the radioactive isotope of sulfur? S35. Okay? Normal one is S32. This is first experiment. Let us come to the experiment number 2. In experiment number 2, the culture medium used was same. E. coli was placed here. Essential ingredients were also used. But only thing which was different from experiment number 1, here the radioactive phosphorus was used. That means radioactive isotope of phosphorus was used, which is P32. Here, which type of radioactive isotope was used? S35. Which type of radioactive isotope of phosphorus was used? Which isotope was used? Radioactive isotope. That is P35. Okay? Once there was a question posed by, I don't remember actually, by some board of the country, I don't remember actually. Why they used radioactive phosphorus and radioactive sulfur? Why didn't they use the radioactive nitrogen, isotope of nitrogen? The simple answer to this question was that they were searching for the material, which is the genetic material. Whether it is a DNA or whether it is, protein. Okay? Only sulfur and phosphorus were two isotopes fit for this. Nitrogen is present both in proteins and in DNA. If they would have used nitrogen isotope, the purpose would have not been served, because they were searching for a material which one of them would have, in which one of them would have been absent. Okay, I am telling you that they used sulfur isotope only because sulfur is present in proteins, not in DNA. They used phosphorus isotope, radioactive isotope, because phosphorus is present in DNA, not in proteins. Is that clear to you? Now, in first experiment, we have E. coli, we have, culture media, and in addition, we have radioactive isotope of sulfur, that is S35. And in another experiment, we have E. coli, culture media, E. coli is there, but there is radioactive isotope of phosphorus, that is P35. And then they allowed bacteriophages to infect this E. coli. Now came the bacteriophage. Okay? This infected this E. coli in the culture medium. Okay? Another came the bacteriophage. Let me briefly tell you how a bacteriophage infects its host. That means bacterial cell. Number one, this is a multi-step process. In first step, bacteriophage attaches to the bacterial cell. Okay? For attachment, it has structure of tail pins and tail fibers. Okay? When this attaches with the bacterial cell, bacteriophage, it breaks down bacterial DNA. Okay? If I will tell you, this is a bacterium, E. coli. Now this is a bacteriophage. It has attacked this bacterial cell. Here E. coli has its own DNA. Virus has its own DNA. Once this virus, bacteriophage, gets attached to the bacterial cell, it breaks down the cell wall first because of enzymes present in tail fibers and tail pins. Then it breaks down the bacterial DNA. Okay? Now, it's the first step. In next step, the viral DNA, which has entered into the bacterial cell, right, and broken down the bacterial DNA. Now, this viral DNA replicates. It forms many copies of it. Okay? During replication, part of the bacterial DNA gets incorporated into the viral DNA. This is second step. What is second step? In second step, once this bacterial viral DNA is injected into the bacterial DNA, into the bacterial cell, inside the bacterial cell, viral DNA replicates, and at the same time, this bacterial DNA is broken down, and during replication, a part of bacterial DNA gets incorporated into the viral DNA. Okay? In third step, bacterial cell lysis, it breaks down. Okay? And when it breaks down, bacteriophages are released. And these bacteriophages in the fourth step, which are released, these carry DNA, which has bacterial DNA also incorporated into it. And the same bacteriophages, when they attack another bacterium, they transfer the DNA. If I will repeat the definition of transduction, I had told you that when DNA is carried, fragments of DNA are carried from one bacterial cell to the another bacterial cell by bacteriophages, that's known as transduction. Same thing has happened here. How? This virus, bacteriophage, T2 bacteriophage, attacked this bacterial cell. During this attack, viral DNA passed into the bacterial cell. And this viral DNA started replication. During the process of replication, part of the bacterial DNA got incorporated into the viral DNA. Okay? Now the bacterial cell broke down. It was broken down. Lysis of the bacterial cell took place. New bacteriophages were released. Okay? These bacteriophages now attacked and infected another bacterial cell, and they transferred this bacterial DNA to the another bacterial cell. This is bacteriophage. This is transduction. Okay, now come to the point again. What Harshey and Chase did, they grew bacteriophages. Which bacteriophages? T2 bacteriophages. These bacteriophages, even for that matter viruses, they don't have a high-graded metabolism. Their only metabolism is to, multiply, form copies, nothing more than that. Okay? Now, two culture media were prepared by Harshey and Chase. In culture media, two culture media, only thing which was different was that in one, radioactive sulfur was used. S35. And in the other, radioactive phosphorus was used. Okay? Phosphorus is present in DNA, while sulfur is present in proteins. Sulfurs don't possess phosphorus, and DNA doesn't possess sulfur. What I am telling to you. This is very simple, but this is very complicated. And this is very important from the examination point of view. You just learn it very seriously and very keenly. If you don't get things, if you don't understand things properly, you just pass the degree. Okay? You can go reverse. You can just repeat the things, revise the things, so that you can get it easy. Though it's not so, tough to understand, but it's confusing. But on the other hand, it's very important from the examination point of view. Because Harshey and Chase have given the best possible, world accepted, experiment which has proved once and for all that DNA is the genetic material. Though Griffith, Avery, McCarthy, they do perform experiments, but their experiments were not so much reliable. Many, scientists across the world doubt their, experiments. But this experiment was the only experiment which got wide acceptance worldwide. Everybody was convinced. And hence then, from 1952, it's present, up to the present, time, it is universally accepted that DNA is the genetic material. Okay? Now, when they grew in experiment number 1, they grew bacteriophages. Right? Bacteriophages in a culture medium. This is a bacteriophage. Okay? Suppose this is bacteriophage. It has a head and tail. Okay? It was grown in a culture medium where there was radioactive sulfur. Where this radioactivity will be incorporated? Obviously, it will be incorporated into the proteins. Where is the protein? Protein is in the protein coats. Protein coat. Now, see up this experiment. This is head and tail. Okay, of a bacteriophage. Recently formed. Multiply. Okay. Here, it has been grown in a culture where the source of, phosphorus was a radioactive source. It was not normal isotope. It was a heavy isotope. It was a radioactive isotope. Where this radioactivity will be incorporated? Obviously, it will be incorporated in the DNA. Okay? Now, we have two types of phages which were grown in two experiments by Harshey and Chase. One type of phages which were grown in experiment number 1 have radioactivity in their protein coat, right? Whereas other type of bacteriophages which were cultured, which were grown in, experiment number 2, they have radioactivity incorporated into the core. Into the nucleic acid. Into the DNA. Because here in bacteriophages, DNA is the nucleic acid. Okay? Now, two types of bacteriophages. Where in next step, allowed to to infect normal E. coli bacteria. Now this bacteriophage, this will infect E. coli in a normal medium. This will infect E. coli in a normal medium. Okay? This bacteriophage, this will infect E. coli in two more samples. In sample number 1, this bacteriophage, this is a unique bacteriophage because it has radioactivity in its protein coat, and part of the protein coat. Whereas in another sample, or you may call it an experiment, this bacteriophage has radioactivity in DNA part, nucleic acid. Now see what happened. When this bacteriophage infected E. coli, okay, it incorporated its DNA into the E. coli. Okay, this bacterium also, it incorporated DNA into its, bacterial cell. Injected DNA. This bacteriophage, which is a radioactivity in protein code, or you may say capsid, this bacteriophage, which has a radioactivity in DNA, it also injected its DNA into the bacterial cell, that means E. coli. This also injected its DNA into bacterial cell. After going steps, first it got attached, then it injected its DNA into this, bacterial cell. Then the bacterial cell, got broken down. Then the virus inside the bacterial cell multiplied, and new false particles were formed here, and in the same way in this experiment. After observation, after analysis, after analyzing, it was found that in sample 1 or experiment 1, where new bacteriophages didn't show any type of radioactivity. Why? Because the radioactivity was present in the protein coat. In the other sample, the radioactivity was observed in the new bacteriophages. Why? Because the radioactivity was present in the DNA of the infecting bacteriophage. So, with these two samples or experiments, it was clear that DNA is the genetic material. And this proved to be the worldwide accepted experiment to show that DNA is the genetic material. Before ending this video lecture, I may just repeat once again that last two steps, two samples. We had cultured bacteria, we had culture, Harshey and Chase had cultured, bacteriophages, two types of bacteriophages. In one type, the radioactivity was present in the capsid or core, that means in proteins. And in other type, the radioactivity was present in DNA. When both these bacteriophages infected E. coli, fresh batch of E. coli bacteria, the phages, newly formed phages, showed radioactivity only in case of that sample in which the radioactivity was present in DNA. Whereas in the first sample, in which the radioactivity was only in the protein coat of infecting bacteriophage, they didn't see any type of, radioactivity. And this was discovered that, in this way, this was discovered, proved for once and for all that DNA is the genetic material. Thank you. In our next lecture, we will discuss conjugation. Thank you.